So at first, what is gram staining? So can anyone tell me the definition? Okay. So gram staining, as you know, it is a differential staining technique. Okay. So is a differential staining technique through which we can, it is mainly used to differentiate between two types of bacteria, that is gram positive and gram negative bacteria. Okay, on the basis of the structural differences that is present in their cell wall. So these are the different staining technique. In the first practical class, I guess you have learned about these different types. That is simple staining, negative staining, differential staining. It has gram staining and Zn staining, and the fourth one is special staining. Okay. So can you give me an example of any microbe that shows negative staining? You have drawn also, and you have drawn wrong. Cryptococcus in India ink stain. Okay, so here we will be learning about the procedure, the theory, that is the principle of gram staining, and the examples of bacteria showing, the examples of bacteria that are gram positive, either gram positive Cocci, Bacilli, gram negative Cocci and Bacilli. So, principle of gram staining. There are various theories. Among them, there is the pH theory and the cell wall theory. From both of this, the cell wall theory is more important. Okay, so if you are asked in your viva to tell the principle of gram staining, you will mention about the cell wall theory. So what does the pH theory say? It says that the cytoplasm of gram-positive bacteria is acidic in nature, so it retains the primary stain, that is our crystal violet. You will learn in the subsequent slides that crystal violet we use as a primary stain in the gram staining process. Okay, so it retains the primary stain for a longer time, and in the second step, when we put iodine that serves as a mordant, and it forms a diiodine complex glycan layer, and with tight cross linkages, and thus it acts as permeability barrier and prevents the crystal violet loss. And gram negative cell wall have a thin peptidoglycan compared to gram positive, and it also has a lipopolysaccharide layer. So in the third step of gram staining, when we pour the decolorizer, it it dissolves the lipopolysaccharide layer, and thus it creates large pores and allows the escape of the diiodine complex. Now, who developed gram staining? Scientist name you can know, Hans Christian Gram. And as in the definition I told, its purpose is to differentiate bacteria into two groups, gram positive and gram negative. So what are the components of gram staining? It contains 4 components. Primary stain, it can be with crystal violet, gentian violet or methyl violet, and gram iodine that acts as a mordant. So what is mordant? Can you say? Okay, so mordant is basically a binding or fixing agent. Okay, I have told you in the principle that our gram iodine acts as a mordant. It helps in fixing the primary stain, that is our crystal violet, with the cell wall more tightly. Okay, then after this you have to air dry the smear that you have made and heat fix it. And now the 4 steps of gram staining will begin. So step 1, that smear you have prepared, you have to pour the primary stain, that is our crystal violet, then keep it for 1 minute and rinse it with water. Second step is pour the mordant, that is our gram iodine, keep it for 1 minute and then rinse it with water. Third step, pour decolorizer, that is here we will be using acetone. Okay, keep it for 2 to 3 seconds, then rinse it with water. And our fourth step we will be pouring safranin, keep it for 30 seconds, then wash away. Then we will be putting oil immersion and view it under the 100x power lens in microscope. Yes. Under microscope, gram positive cocci will be presenting like this, and gram negative bacilli will be presenting like this, pink color rod shaped structures. Okay, see, these are gram positive cocci, violet colored clustered structure, gram negative cocci, pink color, gram negative bacilli, pink colored dot shaped, and gram positive bacilli, purple colored rod shaped. In Viva, you will be asked to say the examples of gram positive cocci, bacilli, gram negative cocci, bacilli. Okay, so these things you have to remember. Okay, that's it. So now you can go for the practicals. We will be doing today the procedure.
2 people at a time, okay? Be careful, there is a smear, okay? So there is a smear. You can see now that the smear is fixed here. So you guys know how to hold a slide. You will hold it like this, okay? So who wants to go first? 2 of you can go first. After that we'll do it for the rest of you. You will go first. Who wants to go first? You want to go first? Okay. So come here. First of all, this is a staining rack, you guys know. So put the slide in the staining rack. We'll be giving you full hands on, okay? Do you have the watches?
Yes.
Okay. All of you have watches, huh? So make sure you keep the time. Remember, the time starts from the first drop that you drop, okay? Take the thing. Take the thing and make sure you squeeze, okay? Because sometimes the liquid, for example, the crystal violet may not come out if you don't squeeze it hard enough, okay?
You have to cover the entire
No, you have to flood the smear, not the slide.
this much only.
This much only. Our talented lab technician has already encircled the area that you need to cover it, okay? So you need to cover only the encircled area with the smear, okay? So we will start, and what is the first step?
Primary stain first for 1 minute.
For 1 minute, okay?
1 minute, then we will rinse it up with
Rinse it up underwater. How do you rinse it underwater?
sliding position.
You will hold it with the thumb and the middle finger, and you will direct the water through your index finger into the slide, not directly on the slide, otherwise the whole smear will get washed away, okay? Okay, you've got the slide now, okay? So start. Let's see. That is up to you, okay? Push, squeeze, squeeze, squeeze. Good, I think you are done. You are also done. We're gonna wait for 1 minute, okay? Okay, so remember, you should also check whether the tap works or not, okay? The tap works, but it's like it has some problems here, na?
Flows.
The flow is, so we'll hold the slide like this, okay? And we will look at my hands. You will hold the slide like this, and you are gonna use the index finger to direct the flow of water, okay? You will hold it like with the thumb and the middle finger, okay? I think you can also open your. Don't move it too much. Just open it a little bit to see if your tap works or not. It works now, okay. Close it down. If it's 1 minute, then you should wash. Okay.
So 1 minute.
Okay, start. Open the tap a little bit. Okay. Take the slide, hold it using the 2 fingers, and let the water drip. Just slant it a little bit. Yeah, slant it a little bit. Put the index finger down.
Achha.
Come from here.
Okay.
I think that's enough. Don't do it too much. I'll put it on the staining area. She's doing it perfectly, but stop it. Otherwise, you're gonna wash away the whole smear. Okay, now put it here. Now, what is the second step? Mordant. Okay. So we're gonna take the mordant, and we'll flood the smear for how much long? Mordant for 1 minute. Okay. 1 minute. Place the mordant. There. There's no issue. Flood it. Flood the whole thing. Okay, bas. It's enough. So you're gonna wait for 1 whole minute again, okay? What is the third step? You are doing this thing wrong, okay? What you have to do? Do we have extra smear? Dr. Nakshatra, do we have an extra smear right there? This is how you do, okay? Hey, dekh, jai to na. Put the water down to your fingers like this. Okay?
Sir, can I
Check your own times, okay? No, you are doing it again wrong. Your finger should be above the slides. Otherwise, how you gonna wash the smear?
Sir, 1 minute.
Hold it a little bit. Okay, okay. Do it. Are you checking your time?
So
Is it 1 minute?
1 minute gone.
To wash it now. You have to wash the second. What is the second step? It's to wash now. You're not doing it right again. Okay, that's enough. Hold it at the end. So what is the third step? Remember. Wait.
Then acetone.
Wait. Remember, acetone we have to give it for 2 to 3 seconds. But 2 to 3 seconds is so much of a less time that you don't get enough time to make it wet on the slide. Okay. So you're gonna put the acetone and you're gonna quickly wash it under the sink. Okay. Same way. Wet. You are again doing it wrong. Okay. You have to hold the slide at the end so that your finger rests above the smear. Okay.
It's like this.
Above the smear. You guys get this one?
Yes, sir.
And then you're gonna pour the water on the top of your fingers. Okay. Okay. Now we'll do the acetone. Acetone you're gonna drop and quickly you're gonna wash. Wash it on the tap. Same. Bas, done. Same. You're gonna drop it quickly and you're gonna wash it immediately. Wash it. Otherwise it's gonna go away. Everything is gonna get washed. Okay. Good. What is the final step? No. What is the final step? Final step is called counterstaining. Okay. Final step is a counterstaining. So we're gonna put the safranin there for
30 seconds.
30 seconds. What is the other dye that we can also use?
Sir,
That is
primary stain.
No, in case of counterstaining, what is the other dye that you can already? Safranin and Carbol Fuchsin. Okay. So you're gonna add the safranin. Okay. So you can add the safranin now. The same thing. You're gonna flood the smear, not the whole slide. Okay. 30 seconds. You can start whenever you want. You're gonna squeeze the bottle. Squeeze it hard now. There's no issue. We have enough safranin. Okay, I think that's enough. Is this okay? So you're gonna wait for 30 seconds and then what do we do? What is the final thing that we need to do? We need to air dry it.
Air dried and oil immersion.
Okay? Can tell me. Tell me it in English or Hindi so that the others can also understand. Na? Yeah, it's not tough actually. You need to understand how the things work around here, okay? Because there is a technique to everything. Are you guys keeping a watch on your time? Okay. Is it hard? So wash it na. Otherwise you're again doing it wrong.
Sorry, sir.
It's again wrong. She's doing it right. You're gonna wash it, you're gonna hold it like this and allow the finger. What you are doing, you are doing it like this. So the smear can't get washed, okay? So after that, what is the final step? That is, we have to let it dry, okay? So first what we're gonna do, this is a small trick, okay? Come here, don't do this jiggle motion at the beginning. You're gonna put it at this tight spot around here, okay? You can put it there now, so that the excess water dribbles down. Why are you holding it like this? Is this the way to hold it? No. You hold it on the top now, so you can put it there. You can hold it on the top like this and put it here. Done. So you're gonna wait for the excess water to dribble down, and then after that you can do the air drying, okay? So the weight, we're gonna let it dry a little bit. So you're gonna tell me what is the difference between the cell wall in a gram positive versus a gram negative.
In gram negative.
In gram negative.
Peptidoglycan, we have a lipopolysaccharide.
So gram positive is peptidoglycan layer, which is about 50 to 100 layers in the gram positive one, and in the gram negative, the peptidoglycan layer is about 5 to 10 layers, and there is one
Teichoic acid is, yes, teichoic acid is there. In case of gram positive, there is teichoic acid. In case of gram negative, there is no teichoic acid. Okay. In case of gram positive, there are no porins. There are no periplasmic space. Okay. In case of gram negative, there are porins. There are periplasmic spaces. Lipopolysaccharide, is it present in gram positive? Present gram negative. Okay. So this is the main difference between gram positive and gram negative. So what you need to do, you need to understand the differences. Also you need to memorize their examples. Gram positive, the most common example is Staph aureus, Streptococcus. These are the most common examples, which we can further divide on the basis of what are the tests to divide between Streptococcus and Staphylococcus. So how do you differentiate the different strains of Staphylococcus aureus? You have done a small test, one biochemical test. That is also done in either slide test or a tube test. You know, I know you guys know. Tube में भी होता है, slide में भी होता है. What is that thing? That thing is coagulase test, okay? Coagulase test differentiates between Staph aureus versus the coagulase negative Staph aureus. Okay? And give me some examples of gram negative bacteria. E. coli. Any other? Meningitis. Okay. So these are the Neisseria meningitidis. You're gonna have to tell the full name. Okay. You are not a consultant. You are a student, so you have to tell the full name. In case of E. coli, there is Escherichia coli. There is Entamoeba coli also. So what was your Entamoeba coli is a parasite. E. coli is a bacteria. So how do I know that you have told the right one? So as a student, you have to answer. You have to answer properly. Okay. So the rest of you guys can start now. Both of you can start. You can sit here. You can fix the smear on you at the field where you are getting the optimum coverage. Just drop one drop. I think that's enough. You can take it back. Is this the area you want to focus? Okay. So take it all the way up slowly so that it touches the oil layer. A little bit up. No, it's okay. A little bit up until you find some resistance. Okay. Take it a little bit up. Okay, I think that's enough. Now we can watch through the eyepiece. Use the right eyepiece because the left one has a lot of artifacts. Use the right one. And you're gonna hold the fine adjustment and rotate it anti-clockwise towards you. No, this is a coarse adjustment. Anti-clockwise. This is the clockwise. Remember, listen, this is the clockwise, this is the anti-clockwise. Once you have fixed the smear and you have raised the mechanical stage up to a point, there is some resistance. You're just going to use the anti-clockwise motion to find the bacteria. Okay? Tiash, have you found something? No, sir. You are trying now? Okay. No, condenser is not open. Condenser is like up or down. Will the location be at the highest or the lowest?
Sir, highest.
Highest, okay. And the diaphragm will be open, fully open. Okay. So you can do now. First check if the diaphragm is fully opened. If the condenser is fully at the top.
Yes, sir.
Is the objective lens placed?
Yes, sir, it's 100.
Is it the 100x or the other? Okay, it's a 100x. Huh? Okay. Okay. So you're going to raise the platform first and adjust the mechanical stage. अपना स्लाइड अपने पास रखिए. मेले में लिखा होता है ना, अपनी सामान खुद की सामान की जिम्मेदारी अपने पे है, खुद की स्लाइड की जिम्मेदारी भी आप पे है. समझे? अगर ये आपका स्लाइड तोड़ दे तो भी ये आपका ही गलती है. ठीक है? Is it the right way to see?
I
don't want to break this also.
No, no. What is the use of oil in case of oil immersion?
Sir, the
And
sir,
what is the use of oil in case of oil immersion?
sir, the refractive index of the light and the oil is same, so we can see clearly.
Refractive index of the glass and the oil is same.
Same. That's
So if there is a gap of air between your oil and the lens, will the oil work?
no, sir.
So there is a gap between the oil and the lens. The oil immersion lens is meant to be dipped in the oil. So you need to actually manually take it up at the highest point. At the point you find some resistance. Take it up a little bit more. Look here and take it up. While you are moving the mechanical stage up, always look here and feel for the, okay, I think that's enough. Now I will only move it in the opposite direction. Okay, try to focus it.